Do you ever wonder how scientists determine how closely related two species are, or track how a virus evolves over time? The answer lies in phylogenetic analysis, and in this video, we'll briefly explain what a phylogenetic tree is and then construct one step by step using MEGA12. So a phylogenetic tree is a diagram that represents evolutionary relationships among organisms, genes, or sequences. And branches represent evolutionary lineages, while nodes represent common ancestors. Phylogenetic analysis is the process of using molecular sequence data such as DNA, RNA, or proteins, and then aligning this DNA to reconstruct these evolutionary relationships, which can then be interpreted. This approach is widely used in evolutionary biology, public health, genomics, and even conservation. And with this foundation in place, we'll move into MEGA12 and build our phylogenetic tree step by step. So we begin by selecting the align feature and then go ahead and select the edit/build alignment. So we're going to be retrieving our sequences from a file. Make sure you have your DNA sequences ready in a FASTA format. So we go ahead and import our DNA sequences. So here we have our DNA sequences ready. So usually before building a tree, we must align the sequences so that homologous positions are properly matched. And for that, we're going to be using muscle, which helps us to insert gaps where necessary in order to maximize similarity. So we go ahead and click the alignment feature, then select align by muscle. So select all. So here we see the alignment muscle options, and the gap penalty control how gaps are introduced during alignment. The memory iterations control computational limit and refinement, and the clustering method here, which is the UPGMA, it helps us to determine how sequences are progressively aligned. And so for most analysis, the default settings are usually sufficient. So we'll go ahead and click OK. So here we have our aligned sequences, which will be the foundation of our phylogenetic analysis. So before we do anything further, we have to save the aligned sequences in a mega format so we can be able to re-import the data for our phylogenetic construction. So here we'll go ahead and select export in mega format, and then save it any name you want to use that will then go ahead and save. So we have our aligned sequences saved in mega format. Then you go ahead and click on the phylogeny feature, then construct/test neighbor joining tree. So we will import our saved our saved aligned mega format sequences. So click OK, leave all the options as default, then click OK. Here we have a constructed phylogenetic tree, and there are so many features we can decide to work around and play with depending on what you're looking to achieve and also learn. So for instance, the tree feature helps us to be able to increase the taxon separation since they seem too clustered together. Then we also have the branch feature, the node, the subtree, and then let's quickly check out the layout feature. So currently the tree is in a rectangular format or style. So we also have different styles such as the straight line styles, you have the cladogram, you have the radial style, you also have the circular format style. So depending on what or how fascinating it looks to you. So usually every branch in the phylogenetic tree represents a lineage, and each node represents a common ancestor. So here we have a branch, and then here we have a node. And then sequences that cluster together, such as this, are more closely related, while longer branches indicate greater genetic divergence. So these two sequences here likely share a more recent common ancestor when compared to the others. And also, one tip to know, by default, the tree is unrooted, meaning it does not indicate the direction of evolution. So if we include an outgroup, a more distantly related sequence, we can be able to root the tree. So rooting helps us or allows us to be able to infer the direction of evolutionary divergence. So if we pick a branch here is that can serve as an outgroup or is a distantly related sequence, let's go ahead and root the tree. Click on root. So here we have our rooted tree and in the direction of evolutionary divergence. So like I said, depending on what you're looking to do or, learn, you can just play around the features, and when you're done, you go ahead and save your file or your current session. And that is how we construct a phylogenetic tree.